Yes, this is fine.
Okay.
A very good morning to one and all. It's my great privilege to address you on behalf of Sonandra College of Engineering and Technology and Department of Electronics and Communication Engineering. We are very thankful to AICTE for supporting this short-term training program on emerging trends in nanomaterials for electronics and optoelectronics devices, which was intended to conduct in 3 series of 6 days each. Out of these 3 series, one series last week we have completed. It was on synthesis and functionalization of nanomaterials. I extend a very warm welcome to all the vibrant participants to this 6 days STTP series 2 on fabrication and characterization of nanoelectronics devices. I take pleasure in welcoming our today's resource person, Dr. Mihir Kumar Joshi from Saurashtra University, Rajkot, Gujarat. He is going to talk on the topic hydroxyapatite, a wonderful biomineral for nano device applications. I would like to extend my hearty welcome to our principal, Dr. S. Suresh Kumar, and secretary and correspondent, Dr. S. Ramesh Babu, who gave us full support to conduct this event. Today being the first day, I am very happy to welcome our dynamic resource person. But before that, let me introduce our resource person. I feel very proud to tell about his achievements in brief. Professor Mihir Joshi, he is a professor and head of physics department at Saurashtra University, Rajkot, Gujarat. Professor Joshi's broad area of research is the material science, and some highlights of his research activities, his area of particularly in the crystal growth research and research of nanomaterials, nano and bulk crystals of active pharmaceutical ingredients, grown and characterized various materials such as metallic crystals like bismuth, antimony, nonlinear optical material crystals, metal tartrate crystals by using melt growth, solution growth, and gel growth techniques respectively. The growth inhibition studies are carried out for urinary type crystals, brushite, strobites, calcium, oxalates, etc., and crystal responsible for anthropathy, monosodium urate, monohydrate, calcium, pyrophosphate, hydroxyapatite, etc. As well, cholesterol crystals using herbal formulation. This is vitro study has helped to identify the potent herbal extract for possible therapeutic application. Several nanobiomaterials such as hydroxyapatite and calcium pyrophosphate are synthesized and characterized extensively. The bioactivity, antimicrobial activity, DNA binding efficacy of hydroxyapatite nanomaterials, hydroxyapatite, curcumin, nanocomposite, calcium ion irradiation of collagen, hydroxyapatite nanocomposite, metal ion doped nanohydroxyapatite studies are extensively carried out in collaborative manner. Having expertise in melt growth, solution growth, gel growth, hydrothermal growth, wet chemical synthesis, synthesis by microemulsion, etc. Using various techniques, sir has exposure of various characterization techniques like the powder XRD, single crystal XRD, FTIR, UV visible spectroscopy, impedance spectroscopy, dielectric study, electron microscopy, TZDTA, DSC, has HG measurement, Z scan study, photoluminescence, Raman spectroscopy, and many more. Professor Joshi has published more than 170 research papers in reputed journals of high impact. He has guided 20 PhD students and similarly 20 MPhil students. He received many awards, funded projects from the government agencies, and he has also participated in many events and conferences, national and international, especially international conferences at UK, USA, France, Japan, China, Israel, Singapore, and other countries. He is the reviewer of many reputed journals. His Google Scholar H Index 27. He is having and total citations around 2,258. However, Scopus H Index 22. Sir, I welcome you. I welcome you, sir. Hello, sir.
Yes, sir. Thank you, sir. Thank you very much, sir.
Now session is over to you, sir.
I'm audible, sir. I'm audible, sir.
Yes, sir.
Yeah, sir. Can you hear me?
Yes, sir.
Can you hear me?
Yes, sir.
Fine, fine. Thank you very much. Thank you very much, sir, for very nice. Yeah. Now you can hear me. Fine, fine, sir. And there is a small request. Can you close the box except my view so
Sure,
that
sir. Sure, sir.
the slide is visible completely?
Yes, sir.
Yeah, yeah. Thank you, sir. So that like my box will be in the corner.
Yes,
so
sir.
that
Please go ahead.
the slide will be viewed fully. Yeah. Just thank you, sir. Yeah. Thank you very much, sir, for very nice introduction. And I'm very much thankful to the organizers for the emerging trends in nanomaterials for electronic and optoelectric devices, ETN EOD 2021. And that is from Swaranadhara College of Engineering and Technology, Narsapur, Andhra Pradesh. So I'm very much thankful to the organizers as well as my friend Dubeji for giving me this opportunity to share my views on particularly hydroxyapatite. Now, I come slowly to the topic of my today's talk. The topic is hydroxyapatite, a wonderful biomaterial for nano device applications. Usually, philosophically, there are many definitions. What is happiness? The spiritual leaders, they tell us that happiness is a state of mind. And different people have different ideas about the definition of happiness. But for a common man, happiness means that comfortable living and healthy life. So these two aspects are very important for a commoner, comfortable living and healthy life. Now, for the both the things, the technology is very important. For a comfortable life, we require a very good technology so that we use different gadgets and devices. Those are technologically advanced, so we need a technology for comfortable life. And also for a healthy life, we need a technology. And nowadays, in the pandemic period, we need a technology to live a healthy life. So healthy life and comfortable living, these are the two aspects of comfortable and happy life. Now, in this particular aspect, I would like to introduce myself to hydroxyapatite, a wonderful biomaterial for nano device applications. Now, hydroxyapatite I have chosen because it is having a large number of applications. And because of large number of applications, it is used in different areas. And it is again a very prospective material for nano device applications. Yet only few applications are available for nano device, but major applications are related to biomaterial or biomedical uses. So I will try to introduce first, give an overview of a nanomaterial and particularly hydroxyapatite and uses of hydroxyapatite. And then I will slowly come to the nano device applications, and I will give overview of our research work, what we have carried out since last almost 15 to 20 years in our university. So let me come to the first slide. I think the net is working slowly. So I think till it gets changed, let me introduce that hydroxyapatite is a biomineral, and it is having a large number of properties like osteoconductivity, and again, it is very useful for different applications. So, this internet is unstable.
Excuse me, sir.
Yeah, I think it's not changing that. Let me once again share it
Yes,
because there
yes,
are some problems.
yes, please.
Let me share once again.
Yes, sir.
Again, there is a problem. It's not changing. Let me stop sharing it
Yes, sir.
and let me
And restart the slide
re.
once again.
Okay. Now you can see that. Okay, it's visible.
No, sir. I think it will take some time. Not yet appeared. Kindly check, sir. Screen not shared. And we all know there is climate effect also over there. Sir, please unmute yourself. Yes, sir.
There was a loss of connectivity in between. So let me
Yes.
share once again the slide.
At the bottom, sir, that tab you have to do share screen.
Yeah, that's true. But I'm searching the slide. Yeah.
Okay.
Once again, there was a loss of connectivity. That slide
Yes, sir.
disappeared.
We admit it, sir, because of the climatic problem we have. No issue.
It's slowly opening. Yeah.
Okay, sir.
Now let me share. Yeah.
Yes, sir. Fine.
Now it's
Sir, if you have your
sir,
internet
there is a
weak
small
from that
request.
end,
If you can close the side window. Okay.
pardon, sir.
If you can close it, side windows, then it'll be comfortable. Yeah. Side windows. Only one window should be open. Side window, dialog box,
Yeah. Is this okay now?
if it is possible. No, all the
Sir,
windows are open right now.
from your end, sir,
Your
right hand side, you can see there is option. You can minimize it
okay.
on your screen.
Yeah. Okay. Yeah, I can minimize. Okay. Fine.
Yes, sir. And sir, one more
Okay.
request, sir. If you have your
Yes, sir.
internet connectivity some problematic or slow internet connection, so video
Okay.
you can mute, sir, for a while.
Sorry. Video I can yeah, that's so video is in mute right now. Yes,
Yes,
sir.
sir.
Now I'm coming to the first slide.
Yes,
Uh,
sir.
what is hydroxyapatite? The hydroxyapatite means it's a very important material, as I already discussed. The hydroxyl is an end member plus apatau. Apatau is a Greek word, and the meaning is misleading. So hydroxyapatite, the word comes from this hydroxyl and apatau, and that is why it is known as a hydroxyapatite. And a crystal unit cell with a molecule. So one molecule unit cell is a CA5PO4TOH, and that is for the one molecule per unit cell. And for the two molecules in unit cell, we have a double the formula. So like that, we have a hydroxyapatite. The word came from this Greek word. Now, it is a very important biomineral. So, hydroxyapatite is a mineral found in bones and teeth. So, whenever we smile, our teeth are exposed, and one teeth is having a beautiful enamel. An enamel is composed of hydroxyapatite. So, teeth as well as human, it contains hydroxyapatite. High thermal stability with melting point around 1100 degrees Celsius. And it is a brittle but moderate hardness of 5 Mohs scale. Now, there is a legend regarding the hydroxyapatite. And perhaps due to typical nature, hydroxyapatite, the sage Dadhiji in our Indian legend offered his bones to form a Vajra to kill demon Vritta. So the Vajra was made out of the bones of sage Dadichi, and which killed the demon Vajra. So hydroxyapatite and some composite might have been in the Vajra weapon used by Indra. Now we come to the crystal structure of hydroxyapatite. There are two kinds of crystal structures are found, and depending upon the stoichiometry and pressure. So one is the hexagonal and another is monoclinic one. So this is the hexagonal structure and this is monoclinic structure. And the monoclinic unit cell is obtained from the hexagonal one by doubling the B lattice parameter with an alternating arrangement of anion chain. So these are the references from where I have taken the unit cell parameters. Now we come to the structural significance, because the crystal structure and the unit cell packing is very important for many properties. So 8 out of 20 calcium in the unit cell are located at the calcium site, calcium 1 site, and the remaining 12 calcium are located at calcium 2 sites, and which is also the apex of the equilateral triangle, which is connected to form OH ion in channel. So this is the equilateral triangle formed, and these are the equilateral triangle formed. So these are the depicting the calcium 1 location of ion. This is depicting the calcium 2 ion, and this is depicting only oxygen and hydrogen. So this calcium ion is exposed to the crystal surface, and therefore playing an important role in the physical and chemical properties of hydroxyapatite, such as surface charge and interaction with organic compounds. So stoichiometric structure is usually monoclinic, monoclinic. But over the 211 degrees Celsius, it becomes hexagon. So there is a phase change taking place. Now, there are different applications of hydroxyapatite. So slowly I'm coming to the applications like implant material. So hydroxyapatite is a very popular implant material. Hydroxyapatite is not only biocompatible, but also bioactive material. And it directly bonds or binds to the bone and favor the implant fixation due to the properties like osteoconductivity, non-toxicity, non-inflammatory behavior, and non-immunogenicity. So these are the implant material. This is the tip implant material, and the tip implant is made by using hydroxyapatite. Again, for eyeball implant material, it is used for orthopedic implant and orbital implant for removal of eyeball. So if anyone is having, any patient is having a damaged eyeball, and after surgery it is to be removed, so artificial eyeball is implanted, and that artificial eyeball is made up of hydroxyapatite. So these are the references from where I have taken, and these authors have discussed in detail the implant materials. Now, another important application is again in a biomedical engineering, and that is nanoscience applications. So it is the application regarding the hydroxyapatite scaffolds. So porous hydroxyapatite scaffolds have been reported using polymer replication method, which finds application as matrices for bone tissue engineering for specific release vehicles. So it is a bone tissue application. So bone tissue engineering is a promising area focusing on the development of implants capable of repairing and or substituting damaged or lost bone tissues. So these are the bone tissue engineering. It is a very promising method. So for developing porous matrices, for the specific case of bone tissue engineering, some criteria are required to be fulfilled. For instance, porosity, allowing cell to migrate through the pores, and a good condition for nutrient transport, tissue filterature, and as well as ultimately the vascularization. So all these things are very much required for bone tissue engineering, because the cells are allowed to migrate. It should be the porous one, and it should be non-toxic one. Vascularization for the supplying different nutrients are also required. So these things are possible by using the hydroxyapatite scaffold. So this technique shows the schematic diagram, how the scaffolds are made, and the references I have cited at the bottom of the slide. One another aspect of hydroxyapatite is gas sensor. So we know that there are a lot of industries, they require sensing of gas, and to avoid the hazards, gas leakage hazards. That is why the gas sensing and detection is very important. Hydroxyapatite-based carbon dioxide gas sensors are developed, and the gas sensing is measured in terms of change in resistance of hydroxyapatite thin film. So hydroxyapatite thin films are used, and in terms of changing the resistance due to the OH ion, OH ion is playing a very important role. So the proposed model is based on OH ion of hydroxyapatite, playing a role in absorption of the carbon dioxide and change the electrical properties. So various gas sensors are available, and the total market is of 2.2 billion US dollars. So it is a very promising market for gas sensing. Now, another engineering aspect is fuel cells. Right now, we are searching for alternative, the energy devices to replace the combustion engine. So to replace the combustion engine, we are trying to find out the alternate energy devices. For that, the fuel cell is one of the important energy device. So hydroxyapatite conducts proton as low as 200 degrees Celsius. And this property could be used in the production of an intermediate fuel cell. operating between 200 degrees Celsius and 300 degrees Celsius would provide the advantages of the high and low temperatures. So it is having a high operating temperature, and also it can be operated as moderately low temperature. So the membrane of hydroxyapatite or some hybrid material is a potential mean for producing an intermediate H2O2 fuel cell. It is based on H2O2. Hydroxyapatite consists of OH chain of ions along the crystallographic C-axis in a column formed by calcium triangles and phosphate groups. So these are the calcium triangles and phosphate groups. So this is the channel. It is highlighted over here. This is the channel. And the conduction, the proton conduction is taking place along this columnar channel. So that is why the hydroxyapatite is a promising material for the fuel cell. And this is showing the design of a fuel cell. And the fuel cells are developed for H2O2 based fuel cells. And these are the references who have developed these fuel cells. Another very important biomedical application is DNA separation. Now, I'll be touching little bit biology also because it is a very, very important biological material. Hydroxyapatite is attracting large number of biologists. So the important part of DNA separation is DNA binding with hydroxyapatite is very stable. And the DNA sample can be obtained for archaeological bone also. So people have found out from the fossils of dinosaur, the DNA bound with the fossil, and they've retrieved it. And we have seen the movie in Hollywood, where the people have retrieved the DNA from the fossil of the dinosaurs. So it is a possible thing, because DNA binds with hydroxyapatite in a very stable manner. And hydroxyapatite is a constituent of a bone. And when bone is fossilized, the hydroxyapatite remains stable, and also DNA remains stable. And that is why DNA can be retrieved from hydroxyapatite from the archaeological sample. So that has been done. The adsorption of proteins to hydroxyapatite is a complicated, as it involves both anionic and cationic exchange. The calcium functional group can interact with carboxylate residue at the protein surface, while the PO4 and PO4 ion functional group interacts with the basic protein residues, and that is why the selective interaction is taking place, and that can play a very important role in DNA separation. And here it is shown that the calcium does not interact with amino group, and the calcium interacts with the carboxylate group. And here the phosphorus does not interact with the carboxylate group, because PO4 and C double O ions, they do not interact. Here, the PO4 and amino, they don't interact. And like that, there is a separation. It's something like a tearing apart A and B into two parts. So chemically, we are tearing it apart, A and B, into two parts. So like that, the DNA separation is possible. And this slide is showing that how it is active. And these are the references at the bottom I have cited over here. Another important part is in cardiac stents. Now we know that the cardiac problems are major problems in the world, and in society also, large number of people are suffering from the cardiac diseases, and that is why many people, they require stent, cardiac stent, to have a the blood vessels blockage to be removed in the blood vessels. So the Kiddy and Murphy studied the feasibility of using heat blasting process to put nitinol stent with hydroxyapatite. Nitinol is a shape memory alloy. So it is a nickel titanium alloy, and it is a shape memory It is having a shape memory of that particular alloy. And this method has produced consistent hydrophilic and durable ATP or hydroxyapatite stent coating that has the capacity of incorporating a drug-eluting function. So the stent put in the blood vessel to make them broad, these are made up of alloy, shape memory alloy, nitinol. They're coated with hydroxyapatite. And the Grete blasting method is used. The Grete blasting is a process by which abrasive particles are made to impinge on a component to clean or modified surfaces. So this is the general Grete blasting process, very popular in material science. It is used to coat hydroxyapatite on a cardiac stent. Now let me come to another slide. In catalysis, as a catalyst, hydroxyapatite is widely used because it is having a strong adsorption ability, surface acidity, or basicity, and ion exchange ability. So these three properties are very important for catalyst process or catalysis. One, the strong adsorption ability. So adsorption should be very strong. Second is surface acidity or basicity. So that helps the ion exchange ability. So hydroxyapatite application in oxidation reaction, reduction reaction, and the formation of CC, carbon-carbon, also in oxidation reaction of alcohol and dehydrogenation of hydrocarbons is achieved. And at the bottom, I have given the reference where the hydroxyapatite is used for catalysis process. Now, there are adverse effects of hydroxyapatite. And we have just praised hydroxyapatite for different applications, but there are other side also, darker side of hydroxyapatite. So hydroxyapatite is also responsible for ailment called arthritis, and particularly calcific periarthritis, acute chronic inflammatory arthritis, destructive arthropathy, and soft tissue calcinosis. All these complicated names are there, but in short, we call it as a arthritis. And we know that after age of 60, people are suffering from joint-related problem, arthritis. And in the left-hand side, the picture I've shown, the joints are getting painful, and those are shown with a red highlight things. And again, it shows the X-ray, the knee joints, and as well as the palm joints, fingers, they are getting swollen. So the tiny crystals of hydroxyapatite formed in or around joints can cause inflammations of joints and also the tissue around the joints. Such tendons or ligaments, they have been described particularly as a cause of rotor cuff inflammation of the shoulder. And this inflammation is referred to as a hydroxyapatite crystal disease. So deposition of a small hydroxyapatite crystals through our body fluid at the joints, they are causing the disease. And this is the disease due to the deposition of tiny crystals of hydroxyapatite, and these are very painful. So we have to avoid the deposition of hydroxyapatite. So the reference is given at the bottom. Another adverse effect is hydroxyapatite deposits in cardiovascular tissue are commonly found, and it is important to identify at early stages. Hydroxyapatite is also a culprit in the deposition in the vascular tissues or cardiovascular tissues, and they are again forming a plaque. So recently, the attempts have been made to identify the deposition at early stages so that we can avoid the deposition of the hydroxyapatite in our blood vessels. So recent attempts have been made to selectively detect in the early stage of calcification process using the probe. It is CYHBP19 probe. So it is a fluorescent probe. This new probe makes it possible to study the earliest events associated with the vascular hydroxyapatite deposition at the cellular and molecular levels. So this target selective molecular imaging probe approach holds high potential for revealing early pathophysiological changes leading to cardiovascular disease. So if we early identify and detect the calcification taking place in our blood vessels due to hydroxyapatite, then we can take a prevention precautions and we can detect the problem at early stages. So it is a very important part. Now, one more adverse effect, hydroxyapatite and carbonate appetites are found in urinary stone. So many people in our society, they are suffering from urinary stone problems. And these urinary stone problems are due to the bad water, drinking water. And that is why the many minerals are being deposited in our urinary system and ultimately they cause the urinary stone. Hydroxyapatite is one of the component in the urinary stone. And again, I have cited the reference regarding the hydroxyapatite deposition in a kidney or urinary system. Now we come to the another aspect of nanohydroxyapatite. And the nanohydroxyapatite, they have numerous applications, and particularly in the drug delivery system. So clinically, the hydroxyapatite can be loaded with a drug or radioisotope very quickly within 15 minutes. So that is a very good achievement that hydroxyapatite can be loaded with a drug or a radioisotope very quickly. Because if quickly it is done, we can achieve the benefits as early as possible. Now, chlordronate, loaded nanohydroxyapatite. So chlordronate is a diphosphate which affects the calcium metabolism and inhibits bone resorption and soft tissue calcification. So this is a very important part in our body. So it is a diphosphonate type of compound. So chlordronate loaded nanohydroxyapatite particles have remained biologically active and are able to give selective depletion in the Kupffer cells. Now, Kupffer cells are specialized cells in liver because the liver is working as a scavenger things. It protects us against the bacteria as well as the toxicity. So Kupffer cells are very important cells in liver, and they are the cells very important for the liver function. So they destroy bacteria and foreign proteins. So this method of drug or isotope loading of nanohydroxyapatite is rapid, easy, and it eliminates the need for additional surface modification of nanoparticles. So that is for the liver protection and for the loading of the drug, and it is possible by hydroxyapatite. So I've taken one example, and this author has reported this in 2008. Another application of nanohydroxyapatite is a gene therapy. Nowadays, the people have a fancy for the gene therapy. So in gene therapy, nanohydroxyapatite is very important. Now, uh, this system may prove to be a promising vector for gene therapy of inner ear disease, especially of vestibular disorder. So many times we have a inner ear problem, and that is leading to the vestibular disorder. And this has been studied on chinchilla. It's a type of a rodent. I have shown a figure or a picture of a chinchilla. It's a type of a rodent, small animal. So people have used the this particular rodent for the study. Now, nanohydroxyapatite is used as a new candidate of known viral vectors for gene therapy. So there are viral vectors and non-viral vectors. And nowadays we are aware that the vaccine for the COVID vaccine, there are two types. One is a viral vector, another is a non-viral vector. So for a gene therapy, due to its excellent biocompatibility, so the polyethylenamide nanohydroxyapatite carrying specific genetic material can pass through the incalia intact around window membrane and demonstrates high transfection or deliberately introducing nucleic acid in cells efficiency, and vestibular dark cell system. So the major cellular target of polyethanamine nanohydroxyapatite in inner ear are vestibular dark cells and vestibular hair cells. Now I have shown the vestibular hair cells. So vestibular hair cells are like a small hair. They are made up of hydroxyapatite. And they are the part of our auditory system, and they are auditory receptors. So whenever the sound comes, that vibrates these hairs, and they are generating a standing wave. And that standing wave is detected by our nose and our auditory system. So hydroxyapatite is very important in our auditory system. And the gene therapy is applicable by using the hydroxyapatite. And this research work was done by Wu and his co-authors, and it is reported in 2012. Now, hydroxyapatite is having a good market. So, nanohydroxyapatite was used first time in Japan, and the name of company is Sangi, and they sold more than 50 million tubes containing nanoparticles of hydroxyapatite. When nanohydroxyapatite is used in toothpaste, it forms a protective film on tooth enamel and even restore the surface in damaged areas. So nanohydroxyapatite is a promising material to be used in toothpaste. Another part is a global orthopedic prosthesis market is expected to reach around 19.5 billion US dollars in 2015 and 23.5 billion dollars in 2017. Hydroxyapatite is used for the coating the prosthetic devices in different applications in our human body. And that is why it is a very big market nowadays. Another aspect of nanohydroxyapatite is in a fertilizer. So hydroxyapatite nanoparticles used as a phosphorus nanofertilizer. It enhances soybean growth rate and seed yield by 33 to 20% compared to the regular phosphorus fertilizer. So it is used as a part of the fertilizer. And soybean picture is shown here. The nanofertilizers can release at slower rates, which contribute to maintain soil fertility by reducing transport of these nutrients into a runoff groundwater and decreasing the risk of environment pollution and toxic effect due to over application. So many times we are giving overuse of fertilizer, and the extra fertilizer run off to water, and they pollute the water reservoir. So that is why it is not advisable to use the overuse of the fertilizer. So to reduce that, nanohydroxyapatite is used. So it gives the nutrients in a required manner only. They don't allow the extra fertilizer to run off. Now come to the next slide. Another is hydroxyapatite urea nanofertilizer. So the high solubility of urea molecules have been reduced by incorporating it into a matrix of hydroxyapatite nanoparticles. Hydroxide is a poorly soluble or insoluble, and urea is very much soluble in water. So the biocomposite or nanocomposite is made. So hydroxyapatite nanoparticles have been selected due to their excellent biocompatibility while acting as a rich phosphorus source. In addition, the high surface area offered by nanoparticles allow binding of a large amount of urea molecules. This method reported here is simple and scalable. It's a very simple and scalable method, allowing the synthesis of urea-modified hydroxyapatite nanohybrid as a fertilizer, having a ratio of urea to hydroxyapatite of 6:1 by weight percent. So this is a novel method where urea and hydroxyapatite nanocomposite is used as a fertilizer, because all countries, they require a very good yield of crop using a good fertilizer. Another aspect is the graphene hydroxyapatite nanocomposite. This is a very normal composite, which is having a large number of applications. So the large number of applications of nanocomposites of hydroxyapatite and graphene oxide, and reduced graphene oxide. So all the three are having a large number of applications. And in this picture is the review, which was done by Gang Wei, and that was published in Nanomaterials 2019. So that is having a promising use in bone growth, then cell growth, then as a biosensor, then antibacterial applications, and again in a drug delivery. So all these applications are possible with the graphene hydroxyapatite nanocomposites. Let me come to one more application of that. So hydroxyapatite coated iron oxide. So hydroxyapatite coated iron oxide nanoparticles are used magnetic hypothermia medicated cancer therapy. So the researcher, one S. Mondal, and his associates, they developed the hydroxyapatite coated with iron oxide. So the in vitro hypothermia, that is up to 45 degrees Celsius, was reached within 3 minutes, which shows a very high efficiency and kills nearly all of the experimental MG63 osteocarcinoma or osteosarcoma cells within 30 minutes exposure. So MG63 osteosarcoma cells, these are the cancer cells, and these are killed within 30 minutes exposure. So the photographs shows the hydroxyapatite coated with iron oxide, and that shows iron oxide hydroxyapatite as a larger magnification. And these are the characteristic, and this shows the synthesis method and how hypothermia is used for the cancer therapy. Now, nanohydroxyapatite composite devices. These are used for biosensors. Now, graphene oxide hydroxyapatite modified glassy carbon electrode, and that is used for electrochemical cyclic voltammetry. So, electrochemical cyclic voltammetry, for that you require the electrodes. So, graphene oxide hydroxyapatite modified glassy carbon electrodes are used. So these electrodes are used for simultaneous determination of 4-aminophenol, uric acid, nitrate ions, and graphene oxide hydroxyapatite nanocomposite exhibited synergetic effect with high sensitivity, low detection limits, good stability, and high reproducibility. So all these chemical components can be detected by this particularly modified glassy electrode. And the Lavanya and the associates, they use cyclic voltammetry for the detection of uric acid, nitrate, and other components using as a biosensor. Now, another is the metal matrix hydroxyapatite composite. Now, that reminds me the use of a vajra where the metal and hydroxyapatite can be blended together to form a very high strength weapon. So that is my hypothesis, might be done by the earlier times. So MMC HFP composite. There is a high demand to design magnesium alloy with adjustable corrosion rates and suitable mechanical properties. The application of metal matrix composite based on magnesium alloys is quite promising. And a MMC made up of magnesium alloy as a Z91D as a matrix and hydroxyapatite particles as a reinforcement have been investigated in vitro for mechanical, corrosive, and cytocompatible properties. And these mechanical properties of MMC hydroxyapatite were adjustable by choice of hydroxyapatite particle size. So this is having a large number of applications. And the author, White and his colleagues, they have reported the application in 2007. Now, hydroxyapatite PLLA composite, nanohydroxyapatite, incorporated in poly L lactic acid, so it is abbreviated as PLLA scaffolds, develop a fibrous morphology, which in turns increase the protein absorption threefold over non-fibrous scaffold. So it is making it a more porous structure, and that increases the protein absorption. So the results suggest that the newly developed nanohydroxyapatite polymer composite scaffolds may serve as a excellent 3D substrates for cell attachment and migration in bone tissue engineering. So it is a very much applicable in bone tissue engineering to produce a scaffold material. So nanohydroxyapatite is a very promising material along with the PLLA. Hydroxyapatite PVA composite. Hydroxyapatite PVA composite is a hydrogel. It is employed as an artificial cornea fringe to improve biocompatibility for an firm fixation. So in eyes, the cornea is there after the removal or any operative measures. For a firm fixation, we use a hydrogel, and that is a HAP hydroxyapatite PVA composite hydrogel. And that is used for the fixation between the material as well as surrounding host tissue. So this shows the diagram shows that how hydroxyapatite and polyvinyl alcohol are embedded to form a gel. So this was reported by Su et al. In 2008. Now we come to the bone structure. So bone is a natural composite material having a collagen and hydroxyapatite. So this shows the bone structure. One is a collagen, it's a composite material, and another is a hydroxyapatite. So carbonated hydroxyapatite or hydroxyapatite and the collagen. And this shows the bone structure. And this is the bone. It is having a surface where the HPO4, Ca, calcium, CO3, these different ions are located. And this is forming a hydrated. And in solution, if these ions are there, then deposition is taking place. So this shows the importance of hydroxyapatite in bone. Now, bone structure is again shown over here in much more detail, that the bone structure and the different type of the fibers and bone implant materials are developed. So hydrogel for cartilages, porous sponges for bone, as well as fiber orientation and hierarchical fibers and waves and different type of bone implant materials developed depending upon the mimicking the bone using hydroxyapatite. Now, hydroxyapatite collagen composite. So that is shown by here, the hydroxyapatite and collagen. This is the collagen and hydroxyapatite. So this composite is made up of hydroxyapatite and collagen. Then hydroxyapatite collagen scaffold. These are used. So this is the collagen, this is the hydroxyapatite crystal, and the scaffold are used. So soft and ductile collagen and hard and brittle hydroxyapatite, both properties are used in scaffold. It's one type of composite. And these scaffolds are shown over here, artificially made. And these are the electron microscopic diagram. These are showing the microstructure of the scaffolds. The hydroxyapatite collagen, again, it is a very important part of that. So Goldener and trichrome trained histological sections of non-modified collagen scaffold are shown. So these are the histological scaffolds. So in a molecular laboratory, the histopathological scaffolds are done. So the collagen scaffolds and apatite-coated collagen scaffolds that were loaded with various doses of BMP2. It is a bone morphogenetic protein, and implanted into a cavalier defect of mice for 8 weeks. And the arrow shows that how the defect is regenerated or cured. So the in vivo study shows that bone morphogenetic protein 2 delivery with hydroxyapatite coated collagen scaffolds resulted in significantly greater with non-modified collagen scaffold. And this study suggests that the simple apatite coating on collagen scaffold can enhance the bone regeneration efficacy of BMP2 released from collagen scaffold. So it is used in bone regeneration after the fracture. So this is a very important study. Another is hydroxyapatite collagen implant. So implantation into bone defect caused by benign tumor. So the tumors are of two types. One is benign, another is malignant. So the cancerous are malignant tumors, others are benign tumor. So porous hydroxyapatite collagen was implanted into bone defect in the patella, and although the osteosclerotic wall remained, the final efficacy score was 4 and the regeneration was classified as high. So here the X-ray shows that the regeneration of bone. So this is before surgery. Here you can see that this part is getting fused, and again you can see that it is much more short. So this is a very important application. There are various synthesis methods. So different methods of hydroxyapatite synthesis are available. One is solid state synthesis at high temperature, synthesis in aqueous phase, hydrothermal methods, growth from molten salts, growth in gels. So we have worked growth in gels. So there are several other methods, including synthesis by alkoxides, sol gel, chemical methods, as well as several techniques for obtaining coating of various substrates, such as electrochemical deposition, plasma spray methods, pulse laser deposition, etc. Now, synthesis of nanohydroxyapatite, again, those methods, some of them are repeating with the previous slide, because some methods are common with the bulk hydroxyapatite as well as for the nanohydroxyapatite. So these methods are by the nanohydroxyapatite have been synthesized by different techniques like wet chemical precipitation, solid state reaction, sol gel, hydrothermal method, cationic surface, surfactant template, emulsion liquid membrane, mixed surfactants, microemulsion, reverse microemulsion, sol gel emulsion, flame pyrolysis, hydrothermal microemulsion. And we have used in our laboratory surfactant mediated approach and wet chemical method to synthesize pure and doped hydroxyapatite. So this has been done by our student, Dr. Kashmira Tank, in 2013. Now, this is a simple method, a schematic diagram that how it is done. So one by one, we are adding. So this method is that the Triton X100 is used as a surfactant, calcium nitrate, potassium dihydrogen phosphate, and different other substances are used, additive, like arginine or curcumin or collagen can be used, and surfactant method, surfactant mediated method can be used in a very simple laboratory by using very simple glassware even a small college or a small institute can synthesize this surfactant-mediated hydroxyapatite very easily by using simple glass phase and chemicals. They don't need any sophisticated instruments. Now, curcumin is a phytochemical of popular Indian spice. We call it haldi, which is a member of the ginger family. Turmeric, a common Indian spice for curry, and it is called as a haldi powder. And also it is termed as an Indian solid gold. So it is looking like a gold, and it is having a large number of medicinal benefits. And that is why one author, Bharat Agrawal, he mentioned in his review paper, solid Indian gold. That is why it is termed as solid Indian gold. Curcumin possesses diverse pharmacological properties such as antioxidant, antibacterial, antiviral, antiprotozoan, inhibiting cell proliferation, inducing cell apoptosis, and anti-angiogenosis. And we have developed the curcumin hydroxyapatite composite material. And this scanning electron micrograph as well as TEM, they show that how the curcumin hydroxyapatite composites are formed. So here one can see this is a curcumin and this is hydroxyapatite. Here also the lighter outlines are for the curcumin. The darker are the hydroxyapatite. Here this is curcumin and the darker one is hydroxyapatite. And these are the in vitro antimicrobial activities we have done with the help of professor in bioscience department. So these are the normal microorganisms which are causing different diseases, and the antimicrobial activity of hydroxyapatite curcumin composite is represented over here. So hydroxyapatite is widely used as a biomaterial. If instead of using hydroxyapatite, if we use the hydroxyapatite curcumin nanocomposite, then we have an extra benefit because of the antimicrobial activity of curcumin that helps much more than the pure hydroxyapatite. So, and these are the synthetic body fluid that how the deposition is taking place, and that is curcumin hydroxyapatite composite. We have a published paper in International Journal of Ceramic Technology, and I will come to this SBF part, synthetic body fluid part, little later on. Now, in this slide, we are going to discuss that what is the SBF or synthetic body fluid. The synthetic body fluid studies showed improvement in bioactivity of an implanted sample compared to the pristine one, and the enhancement in the bioactivity of implant means uh, collagen hydroxyapatite composite is explained on the basis of electronic loss and electronic interaction through various processes, making the surface of composite charge and leading to more surface deposition activity in synthetic body fluid. So here we can see that these patches are the deposition of hydroxyapatite through the synthetic body fluid. And we see that gradually the white patches increases. So that shows that more and more the deposition is taking place. And the synthetic body fluid is having a different type of mixture of the salts, which are exactly available in our human body fluid. So synthetic body fluid is made in the laboratory by using different salts, which are inorganic salts. And by using those salts, we can make a synthetic body fluid. And if we dip the pellet into the synthetic body fluid, having a hydroxyapatite, that it increases the content of hydroxyapatite. And that shows that it increases the content of hydroxyapatite and that area spreads. So we will discuss later on that how it happens. So this is the mechanism that how the the surface area increases. Now, apatite formation or inorganic, that particular uh, inorganic synthetic body fluid is taking place, the apatite formation. This apatite is a amorphous apatite. It's not a crystalline one. So amorphous apatite formation of hydroxyapatite is governed by many factors, such as density, surface area, surface composition, and structure. So here, the synthetic body fluid. And here it's shown that hydroxyapatite. Now, hydroxyapatite, the surface is having exposed by calcium, as well as this is a calcium, and it attracts the negative ions on the surface. So here, whenever the negative surface charge, negative surface on the hydroxyapatite surface, it attracts the calcium towards it. Let me go back. So it attracts first the calcium because the negative surface charge on hydroxyapatite attracts the calcium ions. And whenever the calcium ion sits on the hydroxyapatite, then that deposition becomes a calcium rich. Then once it becomes a calcium rich, then it becomes a positively charged, so that the negatively charged either OH or PO4 ion is attracted towards the positively charged surface. And that is why the deposition of calcium, OH ion, and PO4 ion is taking place. And ultimately, the surface is covered with a amorphous apatite on the particular surface. So the process of formation of apatite depends on the negativity of the surface. And here, the negativity of the surface of hydroxyapatite is shown. And that negativity of the surface is due to PO4 3 ion or OH ion, and it constitutes the negative group, while Ca2 positive ion constitutes the positive ion of the hydroxyapatite. So the negativity first attracts the calcium ions from the SBF or synthetic body fluid. Once it is covered with a positive ion, then it attracts the negative ion. And like that, the deposition is taking place one by one, and amorphous apatite is formed. So likewise, that regeneration of bone is taking place by using the minerals. So collagen hydroxyapatite composite. Collagen is a family of highly developed fibrous protein found in all multicellular animals. So hydroxyapatite and collagen are major components of our bone. So 70 to 80% hydroxyapatite and 20 to 30% collagen. The basic structural unit of collagen is called the tropocollagen, and it is the basic structure of. Now we have done the ion implantation on the collagen hydroxyapatite by using facility at IUC New Delhi, IUSC New Delhi. And these are the powder exit diffraction of pure hydroxyapatite, and the pure hydroxyapatite implanted with the helium ion, and the hydroxyapatite collagen composite implanted with the helium ion. And these are the studies, the AFM studies of the roughness of the surface. And that was reported in PhD thesis by Dr. Bhumika Jogia. And these are the morphological changes taking place. These are shown in SEM micrographs. So morphological changes from needle to spherical one may be due to the energy delivered by implanted helium ion to the surface, which causes rearrangement of atoms within the sample. So this is done. Now, this is the another aspect of hemolysis study. So hemolysis studies is done. Before that, we come to what is hemolysis. Hemolysis is a Greek word. Hemo means blood, and lysis means losing it. So setting it free or releasing blood. So which is the rupturing of erythrocyte or red blood cells and release their content. So red blood cells, whenever they are ruptured, they release their content. So it is released in surrounding fluid, that is the blood plasma. So hemolysis may occur in vivo or in vitro or inside or outside body. So the toxicity of of a material is studied by using one aspect of hemolysis. All synthesized pure and metal ion doped nanohydroxyapatite exhibited non-hemolytic nature and safe to be used in medicine. So any material you synthesize, first that nanomaterial has to be studied for hemolysis. And if it is safe, then it can be used for any medical application. Otherwise, these are dangerous and they will rupture the red blood cells and it is not advisable for any medical use. So we have studied the hemolysis of nanohydroxyapatite and we have reported in Journal of Nanoparticle Research in 2013. And later on, it was again given in detail by Dr. Kashmira Tak in her PhD thesis. Now, biomimetic study. The biomimetic study of hydroxyapatite is widely reported in a synthetic body fluids. The question is that whenever an implant material is put in a body, it is getting the deposition. Why deposition is taking place in our body? Now, deposition is taking place through our body fluid, and body fluids are having different salts, and different salts having different ions, and those different ions are getting deposited slowly, and that deposition is important to study. So that is studied in a synthetic body fluid conditions. So synthetic body fluid was prepared as per the chemical analysis of human body fluid, with ion concentrations nearly equal to those of inorganic constituents of human blood plasma. In this study, the bioactive glass or hydroxyapatite samples are soaked in SBF or synthetic body fluid solution, and their surfaces are observed to be coated with poorly crystallized calcium deficient carbonate containing apatite or hydroxyapatite. Those are similar to bone apatite. So this study is done in a synthetic body fluid that people dip the palate and they try to see that how the deposition is taking place. And deposition is visible in form of a white surface face patches taking place. So slowly the patches increases, that shows that more and more deposition is taking place. So all pure and metal ion doped nanohydroxyapatite samples were studied by this particular method. Now, another aspect is growing crystals of hydroxyapatite. Hydroxyapatite, the crystals are grown in a gel method. This is shown over here on the right-hand side picture. This is the test tube where the gel is formed and the nutrients are poured over the gel surface. And the hydroxyapatite crystals are grown in a microcrystals and they are formed in a Lysagon ring. So these are the periodic deposition of hydroxyapatite particles, and these are in form of a small particle, and these are known as a Lysagon ring. So this is a ring type of structure and disc or ring type of structure, and these are shown in a periodic manner. So gel acts as a conducive medium for hydroxyapatite and mimics the condition prevailing in the soft tissue. So hydroxyapatite disease, as we have noted in our earlier slides, that is due to the deposition of small hydroxyapatite crystals, micro-sized crystals, in our joints, and they become painful. So to mimic that, the soft tissue environment, we have done the experiment with the gel method, and where the deposition of hydroxyapatite is obtained. The several herbal extract solutions have been tested to inhibit the growth of hydroxyapatite crystals in vitro. In vitro means laboratory condition. So we have used different herbal extracts and see that how the deposition can be reduced. So if deposition can be reduced, by that condition, we can reduce the deposition in our body, and that may be important for medical application. So these are the important preclinical trials have been done, and this is a very important for reverse pharmacology point of view. Reverse pharmacology is again a new concept of pharmacology, and that was proposed by Dr. Bhushan Patwardhan and Dr. Ashok Vaidya. That concept makes it very rapid to identify any potent molecule or any particular therapy. So we have tested these type of the fruit juices and herbal extracts for hydroxyapatite inhibition study. Now I'm coming to the concluding remarks. The hydroxyapatite is a important biomineral with many applications. So hydroxyapatite, I have mainly concentrated on hydroxyapatite, its important property as a biomineral. Hydroxyapatite can find nano device applications as sensors, implant materials, drug delivery, etc. So these are the nano applications. Some of those applications are nano device applications. Some of those applications are nano biomedical engineering applications. And some applications are as a sensors, but still it is unexplored. So there are a lot of possibilities for exploring hydroxyapatite for nano device applications. Hydroxyapatite is important material for nano biomedical engineering. Bone is a natural composite of hydroxyapatite and collagen, and that is why that composite has to be explored. Hydroxyapatite based, many composites are used for biomedical applications. Curcumin is a natural pigment having many biomedical applications. The composites of curcumin hydroxyapatite gives better antimicrobial activity and hemolytic nature. So we can experiment with a novel composite, nano composites of hydroxyapatite for antimicrobial applications and even for the gene therapy and discovery. Ion implantation of helium ion improves bioactivity of hydroxyapatite and collagen nanocomposites. We have done the ion implantation, and that improves the bioactivity of nanohydroxyapatite and collagen composite. So these are the areas I have tried to summarize. Some of the results are our own, and majority of are reviewed and work reviewed of different authors carried out. And as I have given in my review, the major applications of hydroxyapatite, its properties, and applications in medical. But other fields are still unexplored. So we can explore those fields. Now, these are our collaborators and contributors. So Dr. Bharat Parekh is now in PDPU Nagar. Dr. Kashmira Tak, she is a faculty in Government Science College. Dr. Bhumika Jogia, she is in Forensic Science Laboratory in Rajkot. Dr. Pawan Kumar Koleria, IUSC, New Delhi. Professor Vrinda Thakur, she is a professor in Biosciences Department of our University, and very brilliant collaborators from medical field, Dr. Ashoka Vedya and Dr. Mrs. Rama Vedya. They are from Kasturba Health Society, Mumbai, and they are the prominent clinical pharmacologists, and they have contributed in allopathy and Ayurveda both. So they are our collaborators for medical applications. And I'm coming to the last slide. Thank you very much. This is our physics department. This is my office. I'm very much thankful to Dubey sir and the organizers for giving me this opportunity. And I'm very sorry that there was intermittent internet loss because of the poor connectivity. Cyclone is expected to hit our area probably today evening or tomorrow morning. So a lot of winds and a lot of the stormy weather is there. So that may have affected the internet connectivity. So I sincerely apologize for poor internet connectivity and loss of video as well as audio. So I'm very much thankful to all the participants for patient hearing, and thankful to the organizers. Thank you very much. Now I stop sharing. Yeah, thank you very much.
Thank you, sir. Thank you. Though some in between we had some problem, but sir, it was good actually, the connectivity we had. So there was no issue. Thank you very much, sir. Even in the bad situation, you were
Thank you very
with
much.
us. So this session, we have completed the first one. Now this session is open for the discussion. So I'm just permitting all the participants. You can unmute yourself and ask questions. Yes, any question?
Sir, I am Srinivasan
Yes, please.
from Kongu Engineering College, Tamil Nadu.
Yeah, yes, sir.
Shall we use any green synthesis methods?
Your
for the
voice
production
is-
of matter? Shall we use any green synthesis
there are
methods?
lots. Yes, yes, it's a good question. There are some papers available. They employ the green synthesis method, and they are using different herbs to promote the synthesis, to exploit the process of synthesis. So many papers are available regarding the green synthesis of hydroxyapatite. Yes, sir.
Thank you, Mr. Thank you.
Yes.
The next question, please. I think, sir, no more question. Yeah. One second.
Oh, thank you very much, sir.
Yeah, sir, one question is there. One participant raised the hand.
Yeah, please.
Yeah, please ask. Yeah, please unmute yourself. I think there is problem with that participant.
Internet problem.
Yeah, there must be problem. Any more question? I think, sir, no more question. Thank you very much, sir. Thank you very much. And it was very wonderful.
Yeah, thank
talk
you very much, sir.
and very informative things we came to know about this new word, actually, what you have used, this particularly composite materials and their applications. So once again, thank you very much. And I'm also very thankful to all the participants because they made this particular session successful. And now we have another session actually at 2 p.m. So I request all the participants to join the link at 1:45 sharp so that we can start the next session without any problem.
Sir, there was a connectivity, okay?
Yes, sir, it was okay,
Dubey,
sir. Little bit it was disturbed, but it was fine, sir. No issue. We can move ahead.
Okay. So there was no disconnectivity in between.
Yes, sir. Excuse me, sir. One person actually, Dr. Wines.
No, sir. There is no disconnectivity.
Excuse me, sir. Excuse me, sir. I'm audible.
Yes, please.
Sir, I'm Dr. M. Vigneshwari from Tamil Nadu. Sir, one more question. I want to ask one question, sir. Sir, can you explain the relationship between arthritis and hydroxyapatite one more time, sir? Please.
I could not follow.
Sir,
can you repeat?
Can you explain the what is the basic relation between arthritis problem and hydroxyapatite? Can you explain one more time, sir?
Yeah, arthritis. Hydroxyapatite.
Because I'm arthritis patient, that's why I asked, sir. Okay.
Yes. Arthritis is due to the deposition of hydroxyapatite and several other minerals or mineral salts in the synovial tissue of our body. And these are the joint tissues where the microscopic crystals are deposited. And I have also explained that the synthetic body fluid, that one can do that particular experiment. And in a body fluid condition, different mineral salts are there in our body fluid, and those are playing an important role. So under the conducive environment, position of different mineral salts one by one is taking place, and they are forming the apatite macrocrystals in our synovial fluid, and those are at the joint tissue. Whenever this deposition is taking place, they impair the joint movement, and swelling and inflammation is taking place, and with acute pain. So there are different approaches how to cure it. So one of the approach we have undertaken, that was by using a different herbal extracts. Now different herbal extracts under the guidance of pharmacologist Dr. Vaidya and Mrs. Vaidya, we have selected the herbal extracts. They have prepared the herbal extracts and they gave it to us. And we used those herbal extracts in vitro in our experiment. And we found that in vitro conditions, they are giving excellent results. Then we gave our results and they did on the animal study. So what they did that in the subcutical part of our animal, they artificially implanted these small crystals, which we provided. And they studied the information. How curing is done, they have studied. That was studied.
Sir, is there any natural medicine for that, sir, or any kind of food?
There are some natural medicines available, but I don't have any idea. But if you can search, you can find out there are some natural medicines available, like Gokshura and other things. They are very much helpful for hydroxyapatite. But in that case, without taking because I advise that you take help of Ayurvedic doctors, because they are expert in that particular field, so they know the age and how to be administered. So I just suggest that you consult the medical people, particularly Ayurveda medical people. They will suggest the particular remedy, which type of drugs are available, because these are the herbal extracts. These are in a powder form, but they are not directly to be administered as a medicine. So to make it into a medicine in a powder form or a drinkable potion, so Ayurvedic people, they have prepared it.
Okay, sir. Thank you, sir. Thank you so much. Thank you.
Yeah, one more participant is waiting. Yes, please.
Yes, sir.
Yeah, Selva Raju.
Yes, please.
Yeah, please unmute yourself.
Professor, good morning, Professor.
Yeah, good morning.
This is Dr. Pradeep Kumar from Coimbatore, Tamil Nadu.
Okay, please.
Thank you, Professor, for giving wide information about this hydroxyapatite and their applications. And, I have been working on this hydroxyapatite for the past 10 years, but application is quite different. It is for the host lattice nuclear waste immobilization. So this material have used for particularly.
I could not follow.
I have used this material for nuclear waste immobilization. Hydroxyapatite. Can you hear me, Professor?
Okay. That's very interesting. Very interesting one.
Yeah. So I have been working on this material for the past 10 years, hydroxy and fluoroapatite. And this material is structurally, right, three-dimensional flexible structure. So it can accommodate varieties of ions into its structure. So, of course, this material is widely being used, but my concern is how far this material is commercially utilized as of now. For example, like tooth enamel and implant material.
Yeah, commercially it is utilized in one of the slides. Yeah, in one of the slides, I have shown you that nowadays the toothpaste, they are having the nanohydroxyapatite. And even the implant material, it's a huge market for implant material. So for the fractured bone implant material, then for the stent implant material. So all the implant materials based on hydroxyapatite, and either it's an alloy coated with hydroxyapatite or nitinol coated with hydroxyapatite. So hydroxyapatite is having a large number of demand. And in fact, I've shown in one of the slides that almost $26 billion per year market demand is there. The thing is that one has to explore that in a commercial manner. And your applications is very interesting.
Yeah,
I think
so
you are doing research along with I think Kalpakam or the
along with the Indira Gandhi Center for Atomic Research. IGCAR Kalpakam.
Yeah. Otherwise, because Kalpakam.
I have studied this leaching analysis of this particular apatite material.
Okay.
And we found in acidic environment some ions like calcium and phosphorus are leaching out during maybe after a month and so on. So in the biocompatible application, if you see that our body fluids will be somewhat acidic in nature, right? So the same, if we apply there in acidic environments, that may come out, no, like the metals will get leach out in the environment. So in that case, how we can say that it is biocompatible?
It depends upon that which metal you are selecting and how hydroxyapatite is processed on that one. So particularly all metals are not selected. Titanium or nitinol, this type of metals are selected. Otherwise, other metals are not compatible for hydroxyapatite. So only thing is that which metal we have selected. So that depends. The stent is made.
Okay, because we have not tried with this titanium and other things. Okay.
Off stoichiometry is more important rather than the stoichiometry. So hydroxyapatite with off stoichiometry is much more applicable than the hydroxyapatite with the proper stoichiometry.
Okay.
Yes, sir.
Okay, Professor. Thank you. Thanks a lot.
Thank you so much. Thank you so much for your interest.
So thank you, sir.
Thank you very much, sir.
Thank you, sir.
Thank you very much, sir.
Thank you very much. And now we'll have second session.
That the internet will, hope that internet will work properly in the afternoon session because of the climatic condition at our place.
Yes, sir. Yes, sir.
Okay. Thank you very much.
We pray for the same. Thank you, sir. Everything will be fine. Thank you.
Yes. Thank you.
Sir, Dr. Dubey, sir.
Thank you.
Dr. Dubey, sir.
Yes, please.
Sir, I'm sorry I could not register yesterday through that because of the network issue. Is it possible?
No problem. You just attend today, and by evening you just put an email to us. We'll provide the link. Again, you can register. Right?
Yeah, I already dropped a mail to you.
Okay, then no problem. You will get response, right?
Thank you, sir.
Thank you. Thank you.